Namaskar, welcome to PK's classes. Today we are going to discuss about the antifungal drug resistance. So before that, let us see the fungal cell. This fungal cell wall consists of manoproteins, glucans, beta-1,3 glucan and beta-1,6 glucan, chitin, and then the cell membrane. So this is the cell wall, and the cell membrane consists of the phospholipid bilayer and ergosterol, and the enzyme beta-1,3 glucan synthase helps in the synthesis of beta-1,3 glucan. So this enzyme is also located across the cell membrane, and ergosterol is the chief constituent of the fungal cell membrane, and ergosterol is synthesized from squalene. And in the nucleus there is nucleic acid, and so the antifungal agents can target the beta-1,3 glucan synthesis or the ergosterol biosynthesis or ergosterol itself and the DNA or RNA synthesis. So you can see here echinocandins inhibit the beta glucan synthase. Amphotericin B, nystatin, these are the pauline antibiotics, and these pauline antibiotics, they bind to ergosterol and form pores through which there will be leakage of the essential nutrients and essential ions, so that will cause the loss of membrane function. And ergosterol biosynthesis, two groups of drugs, azoles and the allyl amines like terbinafine can inhibit. Terbinafine inhibits the conversion of squalene to squalene epoxide. Then azoles inhibit lanosterol to ergosterol biosynthesis. So mechanism wise, we can say pauline antibiotics bind to ergosterol and form pores, cause leakage. Azoles inhibit the biosynthesis of ergosterol, inhibits ergosterol biosynthesis. 5-fluorocytosine inhibit DNA RNA synthesis. Grisans like griseofulvin inhibit microtubular function, so inhibits mitosis, inhibit the polymerization of the tubule into microtubules. Actually they bind to the polymerized microtubules and inhibit spindle formation and hence inhibit mitosis. They bind to the polymerized microtubules, so inhibit the microtubular function. Then allyl amines, they inhibit squalene epoxidase. Thiocarbamates also inhibit squalene epoxidase. Morpholins inhibit ergosterol biosynthesis. Echinocandins inhibit the beta-1,3 glucan synthesis. So accordingly we can classify them, and we can also classify them as systemic, topical, mainly these imidazoles. We can classify them as systemic and topical. Imidazoles, also the triazoles can also be classified as systemic and topical. You can refer to our earlier classes on the antifungal drugs. So now let us discuss the mechanism of resistance. So the modification of the drug target, that is the enzyme, because of the mutation of the genes, is the major cause of mechanism of resistance. So we know this echinocandins, just discussed that echinocandins inhibit beta-1,3 glucan synthase, and that is encoded by FKS gene, FKS1, FKS2, and the allyl amines inhibit squalene epoxidase, that is encoded by ERG1. Similarly, the azoles inhibit the enzyme lanosterol C14 demethylase, which is encoded by ERG11. So these gene mutation can alter the targets and hence can inhibit the actions of these drugs. So azoles cause mutation of azoles resistance occurs because of mutation of ERG11. Choline antibiotic resistance occurs because of mutation in ERG3. Echinocandins because of mutation in FKS1, FKS2, and flu cytosine because of the FUR1 gene. So this during the ergosterol biosynthesis, you can see several genes are there. ERG1 to 27 genes are there, and mutation we have just explained few of the mutations which can cause the resistance to these drugs. Then second mechanism of antifungal drug resistance is increase in efflux, and for that the efflux pumps are encoded by two genes. One is the MDR1 gene, multi-drug resistance 1 gene, and second is the CDR gene, candida drug resistance gene. And mostly because of mutation of these genes, because of these genes mainly, there will be increase in efflux pumps, and that is the cause of the resistance to azoles. So these genes, these two genes, MDR1 gene and CDR gene, are responsible for formation of efflux pumps and efflux of azoles, causing resistance. So these genes are encoded by these transporters are mainly encoded by those genes, and ABC transporter, ATP binding cassette transporter, and MFS transporter, major facilitator superfamily. These two group of efflux pumps are responsible for increased efflux of azoles and hence resistance. Then the third mechanism of resistance is alteration in the cell membrane. So the fungus may cause decrease in ergosterol content due to lack of delta isomerase, and that is the cause of resistance to choline antibiotics. Altered phospholipid and fatty acid composition in candida albicans, and that is the cause of resistance to miconazole. Decreased influx due to lower phosphatidyl choline to phosphatidyl ethanolamine ratio, that is the cause of fluconazole resistance. So altered cell membrane permeability causes the resistance to the azoles and choline antibiotics. Then alteration in cell wall, alteration in cell membrane causes the resistance to choline antibiotics and azoles, and alteration in cell wall can also cause resistance to echinocandins and amphotericin B or choline antibiotics. High chitin content in the cell wall of candida albicans causes resistance to echinocandins. High beta-1,3 glucan content in the cell wall of Aspergillus flavus causes the resistance to amphotericin B. Then there can be defects in enzymes, and mostly it is the cause of resistance to 5-flu cytosine. And 5-flu cytosine, how it acts? 5-flu cytosine, by the action of cytosine permease, by the help of cytosine permease, enters into the fungal cell. Then by the action of cytosine deaminase, is converted to 5-fluorouracil, and this 5-fluorouracil is converted further to triphosphates and monophosphates, which inhibit fungal RNA and DNA synthesis, and so any defect in these enzymes can cause the resistance. And you can see three enzymes. One is cytosine permease. Defect in cytosine permease causes decrease in uptake of 5-fluocytosine into the fungal cell. Then defect in cytosine deaminase decrease the conversion of 5-fluocytosine into 5-fluorouracil, so activation is inhibited. And defects in uracil phosphoribosyl transferase, which converts 5-fluorouracil to 5-fluorouridylic acid, the active form of 5-fluorouracil. So that activation is decreased because of defect in this enzyme. So we can say the defects in enzymes decrease the prodrug metabolism, so activation is inhibited, and that also causes the resistance. In addition to that, there can be biofilms, the biofilms formation, and you can see here because of the Menon expression on the cell wall, this Menon hides the fungus from host immune system, and this is a cause of resistance. And also this biofilm prevents the action of the antifungal drugs, so it protects the fungus from the antifungal drugs. So the biofilms are the communities of hardened cells surrounded by an extracellular matrix, and it is extremely difficult to eradicate this biofilm infection, and this biofilm protects actually the fungus from the action of any harsh environmental condition, including the action of antifungal drugs. So to summarize, the four main category of drugs: azoles, allylamines, Pauline antibiotics, echinocandins, and the fifth one, the pyrimidines, 5-fluocytosine, are the major antifungal drugs, and they show resistance because of different mechanisms, mutation of genes like FKS1 or 2, FUR1, and ERG, and there can be altered steral composition and altered cell wall composition, and there can be transporters like ABC and MFS transporters for efflux of the drug. So, and there can be alteration in the enzymes for the resistance of 5-fluorocytosine. So this is all about the resistance, and we can summarize them. Azoles, because of ERG11 gene mutation, decrease in affinity, alteration in the ergosterol biosynthetic pathway, efflux mechanism by multi-drug transporters. These are the mechanisms for resistance to azoles. Then for Pauline antibiotics, different mechanisms of resistance are ERG3 mutation, reduction of ergosterol content in cell membrane, and high beta-1,3-glucan content in the cell wall. For echinocandins, mutation of FKS1 and FKS2 genes, high chitin content in the cell wall. For flucytosine, mutation of FUR1 gene, defect in enzymes like cytosine permease, cytosine deaminase, uracil phosphoribosyl transferase. So this is all about the mechanism of resistance to antifungal drugs. Thank you.